So welcome everyone to the STEM talk of the 7th Polygent Symposium of Rising Scholars. My name is Jeffrey Aceves. I am a 4th-year bioengineering PhD student at Harvard University, and I will be moderating today's session. The goal of the symposium is to celebrate and showcase the hard work of our Polygent scholars, all of whom have worked tirelessly on their projects over the past couple of months. Today, we will have 4 students presenting on a variety of topics. A fellow member of the Polygent's team will be scoring each student's presentation, which will help us determine prize winners after the event. Please type your questions in the Q&A if you have any questions for each speaker. We will have time at the end of the session for the speakers to answer your questions. Today's speakers are Avi Lekkelapudi, Ethan Chang, Gabriella Felix, and Riya Patel. So that being said, Avi, if you are ready to present, you may go ahead and begin whenever you're ready.
Okay. Okay, so my project was on inner speech and brain-computer interfaces. And my name is Avi Lekkelapudi, and my mentor is Andrew Bender. So brain-computer interfaces are a way for people to control an external device like a robotic limb or computer using only their brain. And the way this is done is, well, many different, the way this is done is brain signals are usually recorded, and many widely adopted BCIs are P300 steady-state visually evoked potentials and motor imagery. But the issue with these common paradigms is that they usually limit speed and are difficult or unintuitive for people to use. As a result, additional speech paradigms have been investigated, such as silent speech, which involves mouthing words without actually saying the sound. But this paradigm is difficult for people who are paralyzed because they can't move their mouth. So a different paradigm is being investigated called inner speech. And my project is on whether or not inner speech can be decoded using machine learning. So starting off, data acquisition. All data was acquired from researchers at an Argentinian lab, and they tested 3 conditions. First, inner speech, where subjects were directed to say up, down, left, or right in inner speech, or just think the words. Visualized condition, where people were directed to move a circle on screen mentally up, down, left, or right. And pronounced speech, where people were directed to say out loud up, down, left, or right, depending on how they were directed. Next, data was actually acquired and recorded using an EEG cap made of 120 electrodes, which basically records the voltage across many different points in the brain, recording the brain signals. And this data was pre-processed using a variety of methods that ultimately seek to reduce noise. So next, signal processing. EEG data is basically a bunch of oscillations at different frequencies overlaid onto each other. So the first way I analyzed my data was using the raw time series data, which involved just time on the x-axis and amplitude on the y-axis. Next, I analyzed it using a spectrogram, which had time on the x-axis, but now on the y-axis, there was frequency, and the intensity of each color basically explained how much power was in that frequency at each individual time point. But both raw time series data and spectrogram have a high amount of dimensionality. And as a result, they need to be used only with models that can handle high dimensionality, such as neural networks. The third way I analyzed my data was using Welch's method, which calculates the power spectral density. The power spectral density basically has on the x-axis frequency and on the y-axis power. And it reduces the dimensionality and noise of the data significantly, meaning it's better for simpler models. Next, electrode and frequency selection. So even with Welch's method being applied, the data was still, it still had high dimensionality. There were still too many features for a machine learning model to analyze. As a result, the data was broken up into individual groups of electrodes and restricted in their frequency. So the data was only analyzed in one of four groups. And these selected frequencies were limited to just these two frequencies. And both the electrodes picked and the frequency, both the electrodes picked and the frequencies were chosen based on literature review. Next, translational algorithms. Okay, so we used 4 algorithms to decode inner speech. And as you can see, the 2 CNNs we used didn't really do that well. They were below random chance, while the simpler naive Bayes classifier and support vector machine both did better than random chance. Now, this seems unintuitive. I mean, wouldn't a more complicated model such as a CNN perform better, given that its complexity would help it decode inner speech better? But turns out the complexity of CNNs actually are a detriment here, because the data set provided is very limited. And because CNNs are so complicated, they need much, much more data to perform well, while simpler models such as the naive Bayes classifier and support vector machine don't need as much data because they're simpler. Okay, so as you can see, inner speech, the visualized condition, and pronounced speech all exceeded random chance on average. You can see that here in this column. However, while inner speech and the visualized condition performed about the same, pronounced speech performed significantly better. And this is discussed here. So inner speech has the potential for BCI use. As you can see, it exceeded random chance, but it's still fairly limited. It can't predict the correct answer consistently. And as a result, further research needs to be done for its use in BCIs. Between inner speech and the visualized condition, they performed about the same. While inner speech performed the best with electrodes centered in the parietal region, while visualized condition performed the best with electrodes in the occipital region. Furthermore, pronounced speech significantly outperformed inner speech, with inner speech having an average accuracy of 28.8%, where 25% is random chance, and pronounced speech having an accuracy of 38%. And the reason for this is pronounced speech had a wider range of frequencies and electrodes that it trained on, with some papers even saying that pronounced speech sees activity up to 150 hertz. Inner speech had performed best with fewer electrodes and frequencies, only performing best with 8 to 12 hertz and with 5 electrodes centered in the left hemisphere. The reason for this is that pronounced speech involves both the semantic meaning of words and motor movement, while inner speech only involves the semantic meaning of words. Given this, having motor movement leads to much more activity in many different regions of the brain, not just in one region with inner speech. Furthermore, having motor movement provides much more information to the machine learning model to analyze. So, future direction. There are a couple things we could do to improve accuracy. The first is data collection using ECOGs rather than EEG. EEGs are non-invasive. You just place a cap on your head to collect brain signals. But the issue with this is that because they're non-invasive, they have a much, much higher noise to signal ratio. There's much more noise in the data, clouding any relevant information. On the flip side, ECOGs are invasive. meaning you actually have to surgically implant them in your brain. But this allows for much more precise data and much lower amounts of noise. Thus, recollecting the data with ECOGs rather than EEGs would help improve the accuracy. Furthermore, data augmentation methods could be used to increase the size of the data set. As I said earlier, the CNNs performed poorly because of how limited the data set was. But with the data augmentation to increase the size of the data set, the CNNs may become a more viable option. Next, transfer learning could be also used. So this method also tackles the issue with the limited data set. Transfer learning involves training the model on a similar task and then retraining it again on the task you actually want to predict. And this helps because by having training it on a different task, you make the training time on the second model less. Next, wavelet transforms. So an issue with Welch's method is that while it decomposed the frequencies, the frequency power was averaged across the entire time window. Wavelet transforms takes the intensity of each frequency at each individual time point, meaning that certain peaks at specific times will be better seen. Thank you. Okay.
Thank you very much, Avi, for that presentation. Our next presenter will be Ethan Chang.
All right. Hi, my name is Ethan Chang, and today I'll be presenting my project, which was a literature review on exploiting the human gut microbiome for novel therapeutics. And this is by me, Ethan Chang, and my mentor was Drew Carson. All right. To give a quick overview of what I'll be talking about today, first I'll be talking about First, I'll be giving an introduction, then I'll move on to bacterial natural products and how those can be used as novel antibiotics. Third, I'll move to live therapeutics. And finally, I will talk about the future of the field and where it is headed. All right, to start off my introduction, I've included a quote by Dr. Tedros Adhanom Ghebreyesus, who's the Director General of the World Health Organization. And he said in 2020, "Never has the threat of antimicrobial resistance been more immediate and the need for solutions more urgent." And this means that although the need for antibiotics to treat bacterial infections remains higher than ever, the discovery and production of new antibiotics has been lacking, which creates a serious problem. Many types of bacteria have developed resistance to commonly used drugs. Thus, researchers have turned to bacteria that live naturally in diverse places to obtain sources of novel compounds. And one of these environments is the human gut. Now, what is the human gut microbiome? The human gut microbiome is a community of microbes that inhabit the body's GI or gastrointestinal system. As you can see in the picture on the right, many bacteria have a symbiotic relationship with their host, which means that we mutually benefit each other. The bacteria provide us with things like food digestion and molecule production, while we in turn provide them with their habitat. And in fact, this relationship is so important that dysbiosis, or a disturbance to the gut microbiome, has been linked to health issues such as obesity and Crohn's disease. And in the gut microbiome, due to competition, bacteria naturally find themselves competing against one another for limited resources. And as a result, through evolution, they have produced natural products to defend themselves and outcompete others. And these natural products are the ones that we as humans can exploit for new antibiotics. Now we'll get into the type of natural products that we can use for antibiotics. First, to discover these molecules, modern scientists use genome mining. And this is important because many natural products are actually hidden within the genomes of bacteria, meaning their genes are not expressed under normal circumstances. And this process starts with genome sequencing, which has been made possible by decrease in cost and more efficient methods that allow bacterial genomes to be sequenced within like an hour, as opposed to back then when it would take even a couple days. And next, after these genomes have been identified, the next step is to search through these genomes for the products, and scientists use bioinformatics tools to search for BGCs, or biosynthetic gene clusters. Many antibacterial products are coded, the genes coding for antibiotic products are located in BGCs, or clusters of related genes, and these BGCs are surprisingly similar among different types of molecules. So this is what allows scientists to search for them. And one type of bacterial natural product is RIPPs, or ribosomally synthesized and post-translationally modified peptides. And these ones are synthesized according to the central dogma of biology, which is basically the DNA is transcribed into mRNA, which is then translated into a peptide chain. And as suggested by the name, these are done in the ribosomes of a cell. Now, the post-translationally modified part means that these peptides, after being synthesized, undergo modifications to turn them into the products that they are. And this could include folding, creating linkages, or even cleaving off parts, such as the leader peptide. And a couple types of RIPPs that have been identified for their antibiotic potential are lasopeptides, which resemble a lasso and have a unique shape that allows them to evade proteases, or the cell's natural defense systems, basically. And this basically makes them deadly against bacteria. An example of this is microcin J25, which was a well-studied lasopeptide produced by a strain of E. coli. Plantazolicin, or PZN, is another type of RIPP, and their linear azoline-containing shape helps them target the membrane and cause lysis of a specific strain of bacteria, B. anthracis. And a third type is lantipeptides, which have a lanthionine residue, and they also attack the target cell's membrane. An example of this is nisin, a widely used food preservative. Finally, sactipeptides, which have a sactonine modification, disrupt target cells by attacking the cell membrane once again. And an example of this is thyricin CD. Besides RIPPs, NRPs, or non-ribosomally synthesized proteins, are another type of natural product. And instead of being synthesized on ribosomes, they're synthesized by proteins called NRPSs. And in fact, many antibiotics that are in use today were first discovered as NRPs, including penicillin. And these NRPs allow for opportunity, as scientists can create hybrid NRPSs to synthesize novel proteins that aren't found in nature. As you can see in the diagram, 3 different NRPSs are fused together to create a new protein. Although the genome mining and bacterial natural products is promising, there are still many obstacles that exist today. That includes many bacteria are still to be discovered. Genome mining techniques rely on already known information with the BGCs, so we may not know if there are different types. Production of bacterial natural products at an industrial scale is difficult. Drugs need to pass through natural obstacles in the body to reach targets such as stomach acid. And finally, the ecological roles of many bacteria are still unknown. All right. And besides genome mining, live biotherapeutics are another way we can exploit the gut microbiome. And live biotherapeutics is basically utilizing gut bacteria themselves to treat diseases. One example is Oxabact, which is a drug developed to treat pH, which is a disease where the body secretes an excessive amount of oxalate, and this can actually cause kidney stones. So in order to get rid of oxalate, the company Oxtera developed a drug using this bacteria, Ooformygenes, which was a bacteria that can metabolize the oxalate. And they put this into a drug that you could take, and these bacteria basically go into your body and break down oxalate for you. And live biotherapeutics has also been used to treat gut microbiome dysbiosis, which, as I mentioned earlier, can be detrimental to the health of the host. And traditionally, this has been treated with antibiotics, but studies have shown that live biotherapeutics are much more effective. And although it may seem intuitive to just put bacteria into a pill, creating live biotherapeutics is actually a process that requires much testing and research. And using Oxtera as an example, the company needed to maintain specific anaerobic conditions to preserve function, and to transport it, the drug was lyophilized, which means it was coated in an enteric coating for protection from things like stomach acid, which could destroy its function. And although there are many bacteria in the gut, there are not natural bacteria to fit every medical need. So researchers have had to turn to engineering at times to achieve the specific results they want. And one way they do this is to get a bacteria to secrete a certain enzyme. And this has been effective against certain metabolic disorders, such as diabetes. As you can see in the graph, a specific study decreased bacteria in mice by a lot using an engineered bacteria. And finally, engineered bacteria can be used to diagnose diseases. This can be done by taking in bacteria that can detect a certain molecule caused by a disease, and this in turn will change the color of the waste of the patient, and this could help decrease the number of serious, sometimes fatal misdiagnoses given out, saving additional lives. And finally, in the future, we will see an intersection of machine learning with scientific research, and we'll see better algorithms to search for more molecules in genomes. And we'll also see directed evolution, which is using known molecules and testing it and creating many variations and testing them against a certain trait to create many variations. And this is repeated over and over again until a molecule that has a certain function is created. In conclusion, genome mining has helped discover promising molecules. Engineered gut bacteria offer an optimistic future. The sheer vastness of the gut microbiome is bound to yield at least some novel antibiotics. Databases will continue to grow with more bacterial genomes. And finally, using a directed approach of synthesizing new molecules will be promising and help us solve the medical needs of tomorrow. Thank you.
Great job, Ethan. Thank you so much for your presentation. Our next presenter will be Gabriella Felix.
Can you guys see my screen?
Yes.
Okay. Good morning, everyone. My name is Gabriella Felix, and today I would like to share with you a proposal to genetically edit the mutated cardiac troponin T gene using CRISPR to lower the incidence of hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy, also known as HCM, is a serious heart condition in which the heart muscle thickens, causing the restriction of blood flow throughout the body. In Figure 1, we have a normal heart on the left side and a heart with dilated cardiomyopathy on the right side. On the left side, the normal heart has roughly similar size left and right ventricles, while on the left side, the left ventricle is a lot bigger and thicker than the one on the normal heart. Though dilated cardiomyopathy and hypertrophic cardiomyopathy are not the same, they are very similar. Dilated cardiomyopathy is when the chambers of the heart enlarge, while hypertrophic cardiomyopathy is when the heart muscles thicken. On the right side, we can see that the heart muscles are thickened, and because of this, over time, the heart will become dilated. Since this is happening, it will make it harder for the blood flow out of the heart. Oftentimes, people with this condition, when the blood flow cannot come high enough, for example, during athletic activities, they can go through something called sudden cardiac death. Initially discovered in 1869, hypertrophic cardiomyopathy is an autosomal dominant disease that often results in sudden cardiac death. This means that an individual has a 50% chance of inheriting this disease. In almost half of the cases, it is familial, meaning that it is passed down through families, while in the other half, it is sporadic. Despite being a common disease, HCM is not always found, as it is typically not shown many symptoms, but a primary indicator of this disease being present is sudden cardiac death. Many genes have found to cause HCM, specifically cardiac troponin T, CTNT, which accounts for 10 to 15% of all HCM cases and is frequently linked with SCD. A missense mutation that occurs in the CTNT tail at residue 92 is one of the leading causes for the HCM mutation. A missense mutation is when a single nucleotide base in a DNA sequence is swapped for another one, resulting in a different codon. Through the gene editing tool clustered regularly interspaced short palindromic repeats, or CRISPR-Cas9, and modify its function. If we look over to figure 2, we can see that it provides a demonstration as to how CRISPR works. This process first begins with scientists introducing the RNA, which will find the spot inside the nucleus where the editing activity should take place. Then the guide RNA will bring in the Cas9, which in figure 2 represents the scissors. The Cas9 will lock onto the double-stranded DNA, where it will cut and unzip the DNA. Finally, following this cut, the new unmutated DNA sequence will be introduced to change the function of the sequence. Throughout this project, I aim to understand the role genetics plays in hypertrophic cardiomyopathy, how gene editing tools can fix genetic mutations involved with HCM, and to propose a novel treatment to reduce the number of people with the CTNT mutation using the gene editing tool CRISPR. In order to accomplish this, I did a literature review on the genetic heart diseases, which led me to find that HCM has numerous targetable mutations. I did a review of gene editing, specifically on CRISPR, and the gene CTNT was identified as a possible target for CRISPR editing, given that it was being linked to high rates of SCD. In order to detect if the CTNT mutation is present, it is proposed that all newborn infants have the CTNT mutation screening included in the standard newborn screening test. Among those found to have the mutated CTNT gene, a treatment is proposed utilizing the CRISPR-Cas9 tool. The Cas9 will splice the mutated DNA, and afterwards, the guide RNA will introduce a new unmutated sequence into the genome, thereby preventing CTNT-related HCM from developing. I would like to draw your attention to figure 3, the effects of proposed CRISPR treatment on CTNT. Here on this graph, we have 2 clusters, one without proposed treatment and one with proposed treatment. In the red, we have the number of individuals with HCM in the United States. On the left side, there are about 700,000 individuals with this disease, while on the right, after the proposed treatment, we have that the number decreased to 595,000 people. There are about 105,000 people that have the CTNT mutation, and of those with the CTNT mutation, 36,563 people die from CTNT-related sudden cardiac death without the proposed treatment. But with this proposed treatment, I aim to decrease the number of CTNT related SCD and those with the CTNT mutation from 100,000 people and 36,563 people to 0. I would like to draw your attention back to the figure 1. In figure 1, we see that the normal heart has similar sized ventricles. And on the left, we see that the heart with a dilated cardiomyopathy has thicker ventricles. Because of this, along with hypertrophic cardiomyopathy, the thickening of the ventricles causes a restriction in blood flow. And as I stated before, over time, they become full and they dilate, which makes it harder for people to pump blood throughout the body. With this, many people die. And because of this, my proposed treatment helps relieve the amount of people that will die in the United States. I would like to conclude my presentation by talking about the main components of this proposal. I want to reiterate that hypertrophic cardiomyopathy is a disease which causes the chambers of the heart to enlarge. And it is also a common and potentially fatal inherited disease. Of the many genes associated with HCM, cardiac troponin T, also known as CTNT, is frequently linked with sudden cardiac death. And the mutation is present in about 10 to 15% of all HCM cases. Through the use of the gene editing tool CRISPR, we propose to edit the faulty section of the DNA with an unmutated CTNT gene sequence. If this treatment is implemented, we predict that over 100,000 people in the US will be cured of HCM, and over 30,000 cases of SCD will be prevented each year. Thank you all for your time.
Hi, Gabriella. Thank you so much. Our next presenter will be Riya Patel.
Hi everyone, I'm Riya Patel, a junior at Monroe Catholic High School in the Bay Area. And today I will be talking about a potential novel treatment for selective mutism through its association with social anxiety disorder. So first I will introduce the topics in my presentation and then discuss in detail both the treatment options for selective mutism as well as its association with social anxiety disorder. And lastly, I will conclude my research with my findings. So first, what is selective mutism? Selective mutism is a rare disorder as it has a prevalence of less than 1% in the United States. It is categorized as an anxiety disorder by the DSM-5, which is the standard classification for mental disorders. Children with selective mutism can typically talk and interact with immediate family members in their home, but will fail to do so in front of others, including their relatives, close friends, and teachers. Selective mutism often interferes with a child's ability to use social communication in his or her life. And because of this, academic and educational impairment can be caused by this disorder, since children typically refuse to speak at school, which hinders teachers from accurately evaluating and assessing their knowledge in subjects. A little bit more about selective mutism. The criteria for selective mutism, according to the DSM-5, is a constant failure to speak in social situations for at least 1 month, and the age that it is most seen is within the ages of 2 and 5 in children. Selective mutism was initially termed and labeled voluntary aphasia and elective mutism to suggest that the child's lack of speaking was a voluntary choice. Now it has changed to selective mutism to show that the child is affected by the disorder in some situations, but not in others, so it isn't a voluntary choice. Okay, so moving on to the treatment options for selective mutism, the two main forms of treatment are pharmacotherapy or medication-based and psychotherapeutic approaches. The most used and effective pharmacotherapy for selective mutism is SSRIs, which stands for selective serotonin reuptake inhibitors, which you might have heard of. And these medications are basically clinically prescribed antidepressants that inhibit the reuptake of serotonin, which allows serotonin levels in your body to increase. And with this increase, SSRIs can help in modulating mood, emotions, and anxiety levels, which are all heavily connected to the symptomatology of selective mutism. The second treatment option is therapies like cognitive behavioral therapy and family therapy, which work most effectively when used side by side with medications like SSRIs. Next, I will discuss the association between selective mutism and a pretty well-known disorder, social anxiety disorder. Selective mutism is associated with comorbid anxiety disorders, such as social anxiety disorder. And I will go over some studies that provide evidence for this association. A meta-analysis found a 75% prevalence of social anxiety disorder in patients with selective mutism from a variety of case studies. And there are also multiple studies that found a 100% comorbidity rate of social anxiety disorder in patients with selective mutism. This table shows the prevalence of anxiety disorders in selective mutism patients. So when looking only at the social anxiety disorder column or the column highlighted in yellow, you can notice pretty high prevalence rates. There are a couple that have low rates, which I looked at and found that they use different diagnostic criteria and assess patients with the disorders in a different way than what is standardly used. Excluding these reports, it is generally accepted that there is an association between social anxiety disorder and selective mutism. This is just a bar graph showing the data listed in the previous slide. And the most important thing to notice is the high prevalence rate of social anxiety disorder compared to the other anxiety disorders. And besides this evidence, there are studies that report similar symptoms in cases of selective mutism and social anxiety disorder. These symptoms include high levels of shyness, distress, and withdrawal. The conclusion based on this association and the treatment options for selective mutism is that serotonin norepinephrine reuptake inhibitors, known as SNRIs, would be a viable novel treatment option for selective mutism. SNRIs are a class of antidepressants that work similarly to SSRIs. They are used to treat depression and anxiety disorders, and images of their chemical structures are shown to the right, and then an image of how SSRIs and SNRIs work is shown to the left. SSRIs and SNRIs are the primary pharmacological treatment options for social anxiety disorder, which is why I concluded that both of them can work for selective mutism as well. This table shows 5 case studies that tested venlafaxine, a type of SNRI, on social anxiety disorder. And most studies found a significant improvement in social anxiety disorder with the use of venlafaxine. And because there is no in vivo test or case study on venlafaxine or any other SNRI on selective mutism patients, I used the general treatment standards to conclude that they would help in the treatment of selective mutism. Generally, treatments used for anxiety disorders should be effective for a range of anxiety symptoms, have favorable tolerability, and have the capacity to reduce present and future symptoms. And because SNRIs reduce symptoms for other anxiety disorders, have low and minimal side effects, and can reduce present and future symptoms for other anxiety disorders, it can be a viable option for the treatment of selective mutism. Based on my research that shows SNRIs' efficacy and reproducibility on social anxiety disorder, SNRIs can help in the treatment of selective mutism in a possibly more effective way than SSRIs have. Lastly, further research should be done on the testing of SNRIs on SM or selective mutism to confidently conclude its efficacy, reproducibility, and safety, as this paper is the first to propose SNRIs as a potential treatment option for selective mutism. So yeah, I would like to thank Lauren Kirk, my mentor, my Polygens mentor, for supporting and advising me in the research, writing, and revision process. I also want to thank Polygens and my family for providing me the opportunity to present at the symposium, as well as use the research I found to create a review paper that I submitted to a journal. These are my references. There are quite a few. So yeah, thanks for listening to my presentation.
Thank you so much, Riya. And now that all 4 presenters have presented, I will now go ahead and read out all the questions from the audience. And if anyone has additional questions, feel free to put them in the Q&A and I will go ahead and ask those. But the first question was directed to Avi. And the question from the audience was, what was the most challenging aspect of your project?
Yeah, I think the most challenging aspect was just finding new things to try. It took quite a while for my model to do better than random chance, and I had to read many, many papers to find a method that would even like help my model accuracy increase by like a percent point.
Okay, thank you very much. The next question for Ethan Chang was, what was the most interesting thing you discovered in your research?
Probably how diverse the gut microbiome is. Like, it's within our own bodies, but there's so much potential, so many different types of bacteria, so much like each bacteria can create like so many different types of things. So it's just like how much is found within this small part of our body.
Thank you very much. The next question for Gabriella was, what next question would you pose to the field to continue your investigations and your studies?
I think I would have to ask how exactly I could make my project a reality. This was a really tough project to do. Looking into hypertrophic cardiomyopathy was something really new to me, and looking into all these genes that could cause this disease was really difficult. And I think that I have created a very well done proposal. And my next question would be, how can I make my proposal a reality?
Great, thank you. And finally, we had a question for Riya, which was, what was the most unexpected thing you learned in your research project?
Probably how effective SNRIs were on like social anxiety disorder. I didn't think a medication like very similar to SSRIs would work in a more effective way, as SSRIs are the most popular option. So I didn't think SNRIs would work better. So yeah.
Great, thank you. And it looks like we have one more question for Gabriella. And that question is, are there any downsides of editing the gene?
I think that a downside could be that maybe editing this specific gene possibly won't do anything. There's also the possibility that something could happen from it, but in order to find out what these downsides would be, we would have to actually edit the gene to see what the outcome would be.
All right, thank you very much. And if there are no further questions from the audience, then I would like to thank those in the attendance and once again congratulate our 4 presenters. I will now end the session, and I encourage all in attendance to check out the other sessions that are occurring at the same time. And congratulations.